Good afternoon, everyone. My name is Jeremy Stuhl, and I have the privilege of being your host today. I'd like to welcome you to today's webinar, Mouse Models with a Chromosome Derived from Wild Mice, presented by Dr. Zhiying Li. Dr. Li is a neuroscientist by training. Her PhD study focused on synaptic transmission and presynaptic vesicle recycling, which was conducted in the lab of Dr. Venkatesh Murthy at the Harvard University. She did her postdoctoral training in Dr. Jeffrey Friedman's lab at Rockefeller University, where she studied cell type-specific hypothalamic networks of metabolic regulation in the context of leptin signaling. Afterwards, she joined Taconic Biosciences, focusing on genetically engineered rodent models for health and disease, supporting genetic science and compliance, as well as scientific and commercial production. She joined Gen Pharmatech US as Director of Life Science and Technology in October 21. Please feel free to ask questions during our webinar today. You can use the Q&A button. Also, this is being recorded and will be accessible from the Gen Pharmatech website in the next couple of days. That said, Dr. Li, the floor is yours.
Well, thank you, Jeremy. It's been a whole year, and it's the last one for 2022. So looking forward to the completion of this year and start a new year. And thank you for hosting me quite a few times. Good collaboration so far. Yeah. Right. So thanks for everybody who joined us today, and appreciate your time, and looking forward to a discussion with you rather than just a monologue. So let me start sharing my screen. Okay, a brief disclaimer. So in today, basically there are four portions of the talk, starting with our usual topic, our inbred mice, which we have worked a lot with and then provide information to our customers and colleagues in the past webinars. And then we'll go into the wild mouse project to let you know what we have been doing and what's our idea behind this particular project. And the third part of it will introduce a couple of strains that has been coming fruitful out of this wild mouse project, and then some application notes on the current status of that project. So starting with the inbred mice that we have been so familiar with, they're great as experimental tools for exploring life science, biology, research and development. They're really well characterized. They're close to human in terms of genetic and genomic information. For the coding regions, they're actually about 85% identical to the human coding sequences in the genome. Their genome are sequenced, so we also have the tools to do genetic manipulation so that we can deep dive into the gene function and protein function and biology behind those coding ACGTs. At the same time, in order for them to be a reliable research tool, their genetic drift has been strictly prevented using breeding, using testing methods, in order to ensure experimental reliability and repeatability or reproducibility. At the same time, the same advantages may actually cast some limitation on these mouse strains. To begin with, they're still rodent. So for us biologists who, fundamentally we want to understand biology from human, they're a tool. They're still different from human beings. There are genetic and genomic differences between different species of the mice, and from species to different species. And then epigenome as well as environmental impact on the genetic information is another big factor. At the very last, as I share with you, the mutations or spontaneous mutations among inbred strains are limited. At the same time, that means they have, for each one of them, they have very limited genetic diversity. And as a whole, their population is also limited in terms of genetic diversity. And this may have a toll in terms of the translatability from preclinical research results to clinical research outcomes. Hopefully I'll share more with you on that perspective. And this is a little something we want to share with you, with the pandemic already in place 3 years with us. We just want to take a look into how our inbred mice has been living in their lifetime versus we humans who has been living under restriction in the past 3 years for different situations or reasons. So mice, inbred mice, for research purposes, they tend to live in a small cage or small space for their lifelong. So for them, this is really feel at home. There's no difference. They're used to it and they adapt to it from generation to generation. People, on the other hand, this is something new. We may or may not like it. We have had behavioral responses to that constriction. So coming back to our topic, thinking about from preclinical research to clinical research, in the field of drug development, understanding or benefiting human health, what has been summarized here is from each step in the process of developing a successful drug, there has many failures accompanying the successes. So preclinical trials, 80% and more are failed trials. And when the successful 10-12% move into clinical stage, again, 80% to 90% fails, and leaving about 12% success among these trials. As you know, a lot of time investment efforts are put into those trials. So in this particular publication, people did a theoretical analysis. What if we can prevent some of the failures that has been observed in the clinical trial stage? Mostly these are the toxicity that was not observed before the clinical trials, but were observed among trial population. So if we can prevent the toxicity, as this paper report, we will be able to achieve a more than 50% success rate for a clinical trial. That's really very encouraging. However, what to do in order to achieve that, that's not a trivial task. So as we, as a mouse provider and the preclinical service provider, we were thinking how we can do to contribute to this theoretical success that we may be able to achieve in the future. So one thing we realized, with the inbred strains of mice, they're very homogeneous. That's the beauty of them. At the same time, they do have limited genetic diversity, which is in contrast to the human population, which is, from small cohorts of human until, later on during the clinical trial stages, the number of patients going in the trials is increasing. At the same time, they're more diverse, different populations, different ethnic groups may be recruited and from different locations around the globe. So we think that's probably one of the limitations that inbred mice has in terms of serving drug development. So in this perspective, we're not the first one to think about it, and there has been many efforts in terms of how can we increase the genetic diversity. So inbred mice, outbred mice, using mice or other species, including rats, rabbits, ferrets, dogs, pigs, even non-human primates. At the same time, when clinical trials are recruiting patients, more and more attention is paying into how to diverse the trial population so that when it goes to public, there will be fewer adverse effects that can impact the utility of the successful drug. So in line with this, we start to think about wild mice because they have been shown to have a lot higher genetic diversity. And at the same, as you can see on my screen in the lower portion of the slide, the team did a whole exosome sequencing analysis from 33 samples of wild mice. These mice were caught by our colleagues and they were analyzed by sequencing. As you can see, among these 33 samples, more than 20,000 SNPs variation were observed. They belong to more than 9,000 genes, and out of these, more than 900 are indels, meaning more than one base pair difference within the specific locus of difference. And then when our team compared those wild mouse samples to either one of the very commonly used inbred strains, BOB-C or B6, you can see the diversity or the difference in terms of SNP variations reduce significantly. So does the number of genes and then the number of indels. So parallel to that, there has been a publication, for example, the one cited here, that wild mice seem to have increased disease susceptibility compared to inbred mice. Currently, that is still being studied by scientists in terms of why. So in our wild mouse project, we are using two methods to introduce the genetic diversity from the wild mouse into the inbred strain, due to the fact that inbred strains are still more stable genetically and easier to breed and keep in a research situation. So there are two ways to achieve this purpose. The first one is called consomic strains. What is consomic strains? Basically illustrated in the two diagrams I'm showing here on the slide. They are the same thing, but showing in a different way. So we can just focus on the one on the left side, which is the donor. For example, a wild mouse is being crossed to an inbred strain, the recipient. So once we get to the first generation, the F1s, half the genome of the F1 is from the wild mouse, the other one is from the inbred strain. It's 50-50 perfect. However, depending on how the future breeding is carried out, you may end up with different outcomes. One of them, if you continue to breed the F1 to the recipient mouse, and then genotyping by doing SNP analysis, you will be able to achieve a consomic strain. What is that? That is, out of the 20 pairs of chromosomes, one of them, one sister chromosome of one of the pairs is from the wild mouse. For example, this one. As illustrated, there's one red chromosome among the total population of chromosomes in this particular mouse strain. So that can be established to become a consomic strain. As you understand up to this point, out of the 20 pairs, only one chromosome, one of the sister chromosome, is a wild mouse chromosome. So in this scenario, all the remaining 19 pairs plus the sister of the red one is still from the inbred strain. So in such a mouse, there has been an introduction where the wild mouse chromosome, one of them, has been introduced to increase the genetic diversity. And other than that, the genetic background of the new strain is still very clear, B6. So this strain can serve as a new type of inbred strain. So far, GEM Pharmatech has done consomic strains on chromosome 1. So we have done, as I've shown you here, 11 new strains on chromosome 1. I'm going to tell you more about that in the later slices. So you may wonder, mouse only have one pair of chromosome 1. How come we can get 11, consomic strains? That is because depending on where you got your wild mouse, each one of them is actually a unique individual. So each one of them can contribute their chromosome 1 to the inbred strain background to become a new consomic strain. So as I've shown you here, in our project, our team actually caught multiple wild mouse from different places in China. And they went through the consomic strain breeding process and established multiple chromosome 1 wild mouse consomic strains. So here, as you can see on the left, upper left panel, these are actually more than 20 consomic strains GEM Pharmatech constructed from wild mouse that are caught from the field. Out of these, 11 were characterized. These are the 11 that we're gonna, bring into our wild mouse project. And for today's purpose, I'm gonna show you 2 of them. One is a YP1, the other one is a ZZ2. There will be more information about them. So what I'm trying to show here on this particular slide is the reason why we're focused on the wild mouse project on chromosome 1. There are 2 reasons. One is chromosome 1 is the biggest among all chromosomes in the mouse genome. So technically, we reasoned if we can do this successfully, we'll be able to do the other chromosomes successfully. So it's a proof of principle in terms of methodology and technical capability. The second thing we wanted to achieve by focusing on chromosome 1 first is that this SNP analysis, as shown you here on the left side, as well as the other part of the slides, is from the SNP analysis, we realized chromosome 1 is enriched in SNPs that are relevant for metabolic regulatory pathways. So the wild mice they have a lot of SNP variations from inbred strain. The majority of these seem to be relevant to metabolic regulation. So we're thinking if we focus on chromosome 1, probably we'll be able to identify some unique genes or genetic mutations that are relevant for metabolic study. So here again, showing you, this is the number 1 chromosome 1 consummate strain I'm going to introduce to you today. It is called the B6 chromosome 1 YP1. So apologize, there is a typo in here. It should be chromosome 1 YP1. So YP is representing the region where this particular wild mouse or the source of the chromosome 1 is coming from. And it has a strain number of D000750. So this mouse is interesting to us in the following ways. I'm going to show you the data. As you can tell, this is a pair of age-matched B6 on the left side, and then this YP1 mouse on the right side. They're still early. They're still young. I think about 12 weeks of age. So as you can tell, very obviously this mouse has a metabolic phenotype, is obese. So showing you here is their body weight. So both strains, B6 and YP1, they are on chow diet, the normal lab mouse diet. But the YP1, they just gain body weight very fast. And then at the same time, their fat percentage out of the total body weight is a lot more than their age-matched counterpart. So before I go on, I do want to let you know, our team did reserve something I'm going to tell you here. So when the mice are younger than 8 weeks of age, they actually don't have this obese phenotype for some reason that we don't understand yet. We're going to explore to understand. But starting from 8 weeks of age, when they are kept on the same chow, they start to gain weight and then become bigger and heavier. So this phenotype is actually quite different from obese mice. If you, anyone in the audience is actually familiar with this particular strain. Obese mice started to become, heavier and more obese, accumulating more fat content in the body early on. The earliest is about 3 to 4 weeks of age, and at that time they actually have a little retardation in terms of their linear growth, but they catch up as they grow older. So moving on to other parts of the metabolic parameters, first thing is they do have hypercholesterolemia, as shown here by cholesterol LDL, HDL measurements. And then on the right side, you can see when these mice became adult, they have fatty liver, very obviously on the top right. And then by histopathology, the obvious fat accumulation or excessive fat accumulation is obvious in their liver. At the same time, you can see, there are, ballooning as well as immune infiltration or inflammation. in the fatty liver tissue. So in terms of glucose homeostasis, these mice, they do show hyperglycemia, but it's not as severe as their lipid profile. So showing you here is oral glucose tolerance as well as IP insulin tolerance tests. They seem to have mild intolerance to both parameters. So we do have the metabolic cage capability. So the mice are subjected to the metabolic cage measurements. Plotting here is the RER, the respiratory exchange rate, which is the indicator of what the energy source that has been consumed or metabolized after the mice eat their chow diet. So as you know, the chow has fat, carbohydrate, and protein. And then the percentage of each one of the macronutrients are actually well defined in the chow diet. So here for a RER greater than 1, that's an indication of carbohydrate is the major energy source that has been metabolized by the animal. So as you can tell, black is the B6, red is the YP1 mouse. Both of them actually have a RER during the dark phase above 1, meaning both of them are actually using carbohydrates as a primary energy source. On top of that, YP1 seems to have even higher preference of using carbohydrates from the diet. So with that, the team want to test whether it's the same thing. This particular strain just have a higher metabolic preference for all the nutrients. So as shown you here, this is a gross morphology analysis to some extent. So these mice are subjected to the normal chow or the chow plus a sugar water, meaning to give them even more sugar to see how they respond to this increased nutrient, and then a high-fat diet, which is giving them more fat than they normally consume. So the top 3 groups are the B6 animals, and then the next 3 groups are the YP1 animals. So if you focus on the green and then the dark blue, they're here. So basically, the YP1 mice, even when they were given more sugar, they don't manifest as obesity, which means their body is capable to consume or metabolize the carbohydrates consistent with the RER observation. At the same time, when they are given a high-fat diet, apparently they just put on a lot more fat, which means their body is not able to consume and metabolize the excessive fat in their chow. So with that in mind, the team looked into gene expression analysis. So here, what they did is taking liver as a example, because liver is the major metabolic organ for mammalians. So taking the liver samples, they did a transcriptome analysis between the B6 and the YP1 mice. As you can tell from the clustering analysis showing on the right side, the B6 and YP1, they have distinct gene expression patterns. And then as shown by our analysis, most of them are actually resembling the DIO when the B6 are given high-fat diet treatment. So our team are exploring more on this perspective, but at the moment, our understanding is given the wild mice are living in their natural environments, they don't have dedicated sources of lipids, and there are not a lot of those in their surrounding. So their major food source is carbohydrates, and that explains why they metabolize carbohydrates preferentially. At the same time, if they are given a constant source of lipids, which is not natural to their natural being, that for them is probably a high-fat diet environment. So that's why they start to show some DIO phenotype. So at the same time, when the mice are on chow diet, our team also observed something quite interesting, which is shown here. On the left side is home cage activity. So basically, there's device in the cages to see, how much the mice are moving during the light phase, which is this portion of the plot, or the dark phase, which is mice typically have more activity during the dark phase. So for B6 mice, you can see the obvious increased activity during the dark phase. However, for the YP1 mice, for some reason that we didn't expect, that they seem to have much reduced activity during dark phase when they were supposed to be very active, searching for food, play around, and explore their surroundings. Parallel to that, what we also see is naturally, without any interference in changing their diet or their eating behavior, they do eat a lot more compared to the B6 mice. So what we observed from this behavior aspect is that for some reason, with the change in their gene expression, they have a lifestyle change, which is they have decreased activity or physical activity, at the same time increased food intake. So this does make us wonder, how is this relevant to human obesity? And we think probably there is a good reason to think maybe these mice, as a research tool, they resemble human obesity better than other strains of inbred mice that we have used and studied before. So now, taking so all the data I've shown you so far is when these YP1 mice on chow diet, and they're different from B6. And there could be, the difference in their environment, in what they used to eat and how much. At the same time, we did want to take advantage of, their accelerated body weight increase accompanied by the other, changes in their metabolic parameters to see how these mice can help us in studying obesity-related metabolic syndrome, for example, NASH, obesity, diabetes, hyperglycemia, and so forth. So one of the traditional models to study all these metabolic-related symptoms is DIO, diet-induced obesity. Basically, is B6 or other inbred strains put on high-fat diet treatment for a long time so that they become obese, accompanied by the metabolic syndrome. So we want to see, one of the limitation factors typically on the DIO perspective is it takes a long time for the diet-induced phenotype to come around. So we were thinking, with the YP1 mice already having accelerated metabolic phenotypes on the chow diet, whether it's possible for them to have even faster metabolic syndrome development if they are put on a high-fat diet. So showing you here in a few slides is what we have seen when they are put on a high-fat diet. So if you just focus on the graph on the left side, body weight, when these mice are put onto a high-fat diet, they become even heavier compared to a B6 that are put on the high-fat diet. Accompanied to that is increased cholesterol, increased LDL. And then these are showing you in terms of time course, when these mice, as you can see the downward triangles, when they are put on a high-fat diet, they start to show much higher cholesterol, much higher LDL in about 3 weeks. This timeline is so much faster than a traditional DIO protocol. At the same time, with the shorter protocol, these mice, as shown you here on the lower portion, in comparison to the B6, when they are given high-fat diet, which is the Western diet, WD, in this slide, they are showing more ballooning. For example, here, the purple panels, as well as fibrosis, which is shown by the red lines in the yellow panels. So these are hallmarks of NASH phenotype. So if you have been a frequent participants in our webinar before, you have seen we introducing the NASH model that we have developed, and that one is also with an effort to accelerate the development of the NASH phenotype, so that particular model can be used for testing potential therapeutics for NASH. So with the same idea, we're thinking, this particular YP1 strain seemed to have a potential to shorten the timeline of NASH development, and then, exhibiting all the characteristics of NASH. So what we did is a proof of principle or proof of concept. We took advantage of one particular drug, TZP, that has been developed and shown to be an effective anti-obesity metabolic syndrome agent. It is a dual agonist for GIPR and the GLP-1R. So we took this particular testing article and applied it to high-fat diet treated YP1 mouse, as you can see here. So we give the TZP in 3 different doses. In the 3 graphs shown you here, the YP1 mice after high-fat diet treatment, they responded to the TZP in a dose-dependent manner. So this data is very encouraging. We're thinking to further develop this and potentially use the YP1 mice as a NASH testing tool. So now I'm going to switch gear to the second chromosome 1 consomic strain I'm going to introduce to you today. It is the chromosome 1 ZZ2. Again, ZZ is representing the region where the wild mouse was originally from, and it has a strain number of 765. So as shown you here, geographically is from a region that is slightly more north in China compared to the YP1 mouse, which is from around the Shanghai area. So if you remember, the chromosome 1, I introduced to you, the variations that we have observed from the wild mouse compared to the inbred strain are indicating metabolic pathways. And then the YP1 strain I've introduced to you and are showing you the data, which is demonstrating, yes, the YP1, due to the presence of the wild chromosome 1, it does have metabolic phenotype. But now here, the ZZ2, it doesn't have metabolic phenotype, but it does have some other interesting phenotype that we want to share with you. So as you can see here, what we observed from this particular strain is a better spatial memory capability. So let me talk you through the data. So what we subjected the mice to, first of all, is Morris water maze. So showing you here is the latency of mice finding the platform where they can escape from the water. So as shown you here, the green represents the ZZ2 strain. The males out of this strain, they were able to find their escape platform faster than their B6 counterpart. As quantitated here, they're also crossing the target quadrant a lot more than the B6 mice. At the same time, we didn't see female showing the same phenotype. So we think there may be a sexual dimorphism going on here. And then quantified on the upper right side is the time they spent in each one of the 4 quadrants. And then number 2 is actually the quadrant where they do have the platform that they can climb onto. So as shown you here again is the male ZZ2 mice. They were able to locate their quadrant, their dedicated quadrant, and then be able to escape than their counterparts. And again, female doesn't have that phenotype. And then here is the speed of the 4 different groups of mice. So apparently they're able to swim just very similar to one another. So After seeing this, we actually subjected this mice to a whole series of behavioral tests that we do have in-house. And showing you here on this particular slide are two of them, open field and forced swimming. So open field on the left side is a test for testing exploratory activities. So mice, as you know, is a nocturnal animal, tend to like the dark, and tend to, because they are prey, so they tend to stay away from the spotlight or the open field. So as you see quantified here, the ZZ2 mouse, both males and females, they tend to stay away from the center than their B6 counterparts. So it seems they don't have desire to explore the wild open field. To probably is to stay away from their predators. And then on the forced swimming test, you can see the graph very easily. They have the capability to swim a lot more than their B6 counterparts. So plotted here is immobility, meaning when they stop swimming, just floating around. Some people interpret this as being hopeless. But we kind of take the same, but we want to say the reverse, which is the wild mice seem to have a lot more desire or greater desire to survive. So they explore, they try to find the way out. And then on the right side is very straightforward, as you can tell. These are videos. There's the B6 mice. Occasionally they will swiggle around, maybe trying to find their way out, but they gave up. That's very obvious. But as you can see, the ZZ2, they continue to try. Whether that's what's the purpose, we wouldn't be able to really find out from the mouse. But given a mouse typical behavior, we do think they're trying to find their way out, to escape from water, which is not part of their natural environment. All right, so parallel to this, we did a little bit more. On the left-hand side is another very interesting behavior testing, which is sort of in the middle of metabolic and behavior, which is a sucrose preference test. So if the mice has a changed feeding behavior or metabolism, they may have a preference for having consuming more sucrose water, which is used in this particular test. At the same time, from the behavior perspective, if an animal prefers to have sucrose consumption, that means they are happy. They're enticed by the good taste of the sucrose. However, if the animal is under a depressive condition, they may not show the same type of sucrose preference as their wild type counterparts. So as you can tell, these four groups of animals, they're all males, B6 male in black, the ZZ2 male in green. And then these two groups, they are living in a normal mouse cage. So that's the mouse space I have in the title of the slide. And then we give the ZZ2 mice a large cage to live in. So what we see is when the ZZ2 mice are living in the normal mouse space, they have reduced sucrose preference. So here we interpret that as a depressive-like behavior. But when they're given back a large cage to live in, so it's the BC, the big cage, the ZZ2 were able to, show the similar type of sucrose preference as shown by the B6 males. Similarly, what we also did, and this is still in progress, is to measure some neurotransmitters or amino acids in the central nervous system. So we have done more than 20, as we told, and then 3 of them showed a significant difference between the B6, which is always the first one, or the one on the left side, versus the ZZ2, which is on the right side. So the first one to show you is from the males. The ZZ2 males seem to have reduced serotonin levels, or 5-HTs, in their CNS, which we think probably has some correlation with this, small space reduced sucrose preference behavior. But at the moment, we don't have a definitive answer for that yet. At the same time, alanine and glutamine showed some difference from the females. We're still trying to understand the data, and then to understand the strain as well. So now coming back to the cartoonish slide I showed you earlier. So inbred strains seem to be just normal in their home cage, or the typical small space. But when the consumic strains, or the mice carrying the wild mouse chromosomes, they seem to have some abnormalities in terms of their metabolism or behavior. So how much these are representing human or resembling human is an open question for us to continue to explore and study. And then when we do, we'll provide more information or share more information with you. So after this point, I've introduced our wild mouse project, focusing on the chromosome 1 consumic strains that we have phenotyped, and then 9 more in the pipeline. At the same time, there are 20 pairs of chromosomes in the mouse genome. 19 of them are non-sex chromosomes. So we, in the wild mouse project, we do have the goal or the desire to introduce the other chromosomes into the inbred strain, as we did with the chromosome 1. However, as you can tell, I'm putting them here again. Using the breeding scheme is a long process. It's quite labor intensive and time consuming. So how to overcome that particular downside? What we were going to do is using a balancer chromosome approach. So let me tell you very quickly what balancer chromosome is. So conceptually, a balancer chromosome, you can take focusing on the right side of the pair of chromosomes. So the one on the left side is the normal one. So it has a directionality, as all DNA and chromosome do. So a balancer chromosome is exactly the same genomic sequence, however, is in the reverse direction. So it's an inverted version of the wild type chromosome. What does this do is, when this wild type and inverted are the pair of chromosome, there is no crossing over or homologous directed recombination that can take place during cell division. So we were able to, so this is another way, as you can imagine, when there's no crossing over, there's no spontaneous mutation, and there's no exchange of the genomic information between the two sister chromosomes. In that way, the inverted one will be able to remain as it is from the start to the end, very similar to the wild chromosome we're introducing into the pairs of chromosomes. So that's the idea of the balancer chromosome. How you can do it, here is the way that our company has done it for chromosome 17. So let me introduce chromosome 17 very briefly. It is not the biggest, as you have heard from me. Chromosome 1 is the biggest one. It has a size of about 90 megabytes, mid-sized chromosome. So what we did in order to create this inverted chromosome 17, we used the LOXP system, and this is how it happened. We targeted the wild type with LOXPs, and then we targeted the part that we want to invert is 94% of the entire chromosome using LOXP and a mutated LOXP. And then using the free recombinase, we were able to switch the directionality of the wild type chromosome. So this study has been published in a paper last year. If you're interested, you can take a look. And here is the reference. So chromosome 17, we have been able to create a balancer chromosome guided consomic strain. Well, I shouldn't say a consomic strain. It is a wild chromosome in the inbred strain. So after this point, our goal is to continue use balancer chromosome to introduce more of the wild type wild mouse chromosomes into the inbred strain genetic background. So as I show you here in this last slide, to sum up, the goal of this particular wild mouse project is to increase the genetic diversity among the lab mouse strains that we can provide to the research community. Overall, with all the 19 chromosomes, we have about 190 strains under development. Our goal is to do the strain generation and then phenotyping, characterizing the phenotype of each one of them and provide them as a cohort to the research community, in particular for people who are interested in using this genetically diverse population of mice to screen for drugs or potential therapeutics for the human population. Right, I think this is my last slide. I'm gonna stop here and thank you for the attention. I wanna put this here just in case if you need our contact information. And with that said, I wanna say thank you very much. I'm gonna stop sharing and then we can see whether we have any questions or not. Jeremy, I cannot hear you.
There we go. Thank you, Dr. Lath, for that great presentation. I had my microphone muted as well as Zoom muted, so unfortunately, so I apologize for that. So thank you everyone for your participation. We do have 2 questions here and we have just a few minutes left, so I think we can get to them. The first question here is, do you have wild mice showing different phenotypes in immunology?
I forgot to mention that. So the reason of chromosome 17 is actually out of the 20 chromosomes or the 19 pairs, chromosome 17 actually has the highest number of immunology related genes. So that's why we target that as the first one for the balancer chromosome project.
Great, great. And then the last question here, and I hope I'm asking this the correct way. So about the wild mice project, which chromosome will be changed next? Chromosome 2 or something else?
Okay. As far as I know, so we have been working on the chromosome 1 for quite a while. As you know, the consomic breeding does take a long time, and we have 11 that we made a decision to characterize. So that's the chromosome that has advanced the most. And 17, we have done the balancer chromosome mutagenesis, or the construction of the balancer chromosome. So that one is going forward, probably second advanced. The next one I know is chromosome 7. That is in the pipeline. How the rest of the genome is ranked, I actually don't know. For people, if you're interested in, and if you have a particular chromosome that you want to be one of the ones that we can work on, let us know. Share that information. We'll discuss with you and see whether we can accommodate in our pipeline development, as well as going back to you to provide the tool for your research.
Outstanding. So that looks like that's all of our questions for now. I appreciate everyone's patience, especially with our bit of technical difficulties in the beginning. With that said, though, I want to wish everyone a happy new year as we round out 2022, and we look forward to seeing you again in 2023.
Great. Thank you so much, Jeremy. And thanks everybody for staying with us. Apologize for the technical difficulty as Jeremy shared. And happy holidays. See you in 2023.
Thank you. Yep, 2023. Thank you, everyone. Take care. Bye-bye.
Bye-bye.